Hello students, so welcome back to the channel. In this video, we are going to study about the cell membrane structure. Now see, cell membrane is made up of phospholipid and protein. Okay, it is made up of phospholipid and protein. And apart from these, some other molecules are also present like carbohydrate as well as cholesterol. Clear? So phospholipid is, now we are going to study about the phospholipid in detail. Now see, what is phospholipid made up of then? The phospholipid is made up of one molecule of glycerol. Okay, this is one molecule, two fatty acids, one phosphoric acid. And to this phosphoric acid, if suppose any other chemical is attached, for example, choline is attached, then we will say this type of phospholipid is known as lecithin. Clear, beta? Now see, what is the structure of glycerol? Your glycerol is CH2OH. This is the glycerol which I am right now drawing it, which is also known as trihydroxypropane. So glycerol, another name is trihydroxypropane. Now I'm attaching the fatty acids to them. The fatty acids means, you can imagine this way, it will be CH3, CH2 n number of times, and N will be COOH. Clear, beta? So I'm just writing it HOOC in the reverse form, CH2 n number of times, and CH3. Second fatty acid is also attached. Now, fatty acid can be saturated hydrocarbon or they can be of unsaturated hydrocarbon. In our explanation right now, we are assuming saturated hydrocarbon. It is a condensation reaction and water molecule is going out to form ester bond. So this is the ester bond formation is going on. Now at the third position, phosphoric acid will be attached. So phosphoric acid means H3PO4. This is the phosphoric acid we have to attach. So see here carefully. This is P, OH, OH, and this OH. And again, water molecule is released. So up till here, it is known as phosphatidic acid. Now I have to add choline to it. Okay, beta? Now we are adding choline. See here. This is choline molecule which is attached. And again you do the condensation reaction. Up till here you are understanding it. I'll just do the labeling, try to understand. This was your phosphoric acid. This is the one is your choline. Clear, beta? This is glycerol. And these two are your fatty acid. Please understand the diagram very carefully so that you can understand how the phospholipid bilayer will be made. Once again, I'm highlighting this is glycerol. These two are your fatty acids. Clear, beta? These are fatty acids. This is your phosphoric acid. And this is the choline part. Just now you imagine this way, you are holding this structure. I'll just highlight it. Please try to understand. We are holding this structure from here. This part is in your hand. This choline part, phosphoric acid, as well as glycerol. This part is in your hand. You are holding this part, okay? Water molecule formation means this bond is there. Water molecule has gone. So you are holding this structure. Clear, beta? Up till here you are understanding it. This structure is in your hand. So it will behave like a head. I'll just show you, wait, just 1 minute. So this is structure which is in your hand. Is having, I think so, how many things it is having? It is having choline. It is having phosphoric acid. And it is having glycerol. What is left is 2 fatty acids are left. Okay, these 2 fatty acids are left. So these 2 fatty acids are coming in the form of a tail. So this structure is known as head. And these are the tails. Okay, these are the 2 tails made up of fatty acid. So head is made up of choline, phosphoric acid and glycerol, and tail is made up of the 2 fatty acid. Let me show you the diagram. Now see beta, try to understand this diagram carefully. Okay beta, this is the choline part. This is the phosphoric acid part. And this is glycerol, to which the fatty acids are attached. So this is the head part. Clear beta? This is head. Focus the diagram, you will focus it here. This is head. Head is what nature? Head is hydrophilic, means it is water loving. And what is tail nature? Tail is hydrophobic. Who is making the tail? It is made by the 2 fatty acids. Okay, these are the fatty acids. In this diagram, we have taken unsaturated fatty acid. That is why bending is there because of the presence of double bond. So fatty acids can be saturated or they can be unsaturated. Now see carefully, the head is hydrophilic and tail is hydrophobic. So it is having both properties in it. So we see such molecules are known as amphipathic molecule. Amphipathic molecules are those molecules which have hydrophilic as well as hydrophobic end. They are having hydrophilic as well as hydrophobic end. Both are present in them. That is why they are known as amphipathic molecule. Clear beta? Up till your concept is clear. Now, see carefully. I told you right now, fatty acids can be saturated or it can be unsaturated. Generally, if they are unsaturated, then hydrogen can be on the same side or it can be on the opposite side. Let me show you. Now see class, there are 2 types of fatty acids are there. One is cis fatty acid, one is trans fatty acid. Both are double bond. Focus the diagram carefully. In case of the cis fatty acid, check hydrogen, both the hydrogen and where the double bond is present are on the same side. Then we call such fatty acid as cis fatty acid. And check in the second one, the hydrogen are present on the opposite side, that is known as trans fat. But trans fat is not good for our health. Is it clear? That is not good for our health. It is responsible for disease that is known as atherosclerosis. You'll be studying in the circulation chapter. Cis fat is actually comfortable. We should have more cis fat. But instead of cis fat, we are having more trans fat. So I think so these are the concepts getting clear. So because of the cis fatty acid, double bonds are there and double bonds are both hydrogen are on the same side. So NIC are produced. So these NICs are actually useful because they are helping to maintain the fluidity of our cell membrane. Now see here, I think so this much is clear. This is head and head is hydrophilic. And these are tails and tail is hydrophobic. So now tell me one thing logically. Let us take one beaker in which I am putting oil. This is a beaker in which this is an oil present. This is oil. And if I put the phospholipid, then I think so you can understand orientation will be. Orientation on top of this will be, I think so orientation will be this way. Tail is facing to the oil side. Is it clear? Tail is facing the oil side. Head will not face the oil side. Clear beta? Now let me give you another example. If suppose in the beaker, instead of oil, I'm putting water. Instead of oil, it is water is there. Then Your orientation will be, head will be towards water side. That is why we say head is hydrophilic, because it is water loving. And tail is hydrophobic, means actually it is water hating, or you can say it can attract towards the lipid, not towards the water. So I think so this orientation is clear to you. Up till here you are understanding it. Now see class, right now I just explain you the diagram, how the orientation will be there. Now see here, this is the water molecule present. Okay, this is a beaker which is containing water, and you know water is a polar molecule. So check carefully the diagram. You're finding head is facing the water, but tail is not facing. Tail is outside towards the oil, means non-polar side. Is it clear? Understood? Now, if suppose oil is only present and oil has to surround this phospholipid, then check oil is also surrounding the phospholipid by making only one layer with the tail. Is it clear? But see one thing, our cell which is having cytoplasm, and cytoplasm is aqueous solution, means water is present. So water is present inside our cell and water is present outside also. Outside and inside the cell, the phospholipid makes bilayer. Understood? Phospholipid will make bilayer. Try to understand this way. This is a cell which is having water, and outside medium is also having water. So how the orientation will be done here? Orientation will be that this layer will be in the form of a bilayer. This way. Clear, beta? Orientation of the phospholipid will be in the form of a bilayer. I hope you are able to understand the diagram now carefully. Are you understanding it? This is the one layer which I'm making it. You can understand this way. And second layer, this is a phospholipid, bilayer is made. So when water is present inside as well as outside, we find that the phospholipid is coming in the form of a bilayer. So our cell membrane is made up of phospholipid bilayer. Now we have to study the fluid mosaic model. Now let me explain you fluid mosaic. Now see, fluid mosaic model was given by Singer and Nicolson. Okay. And what they observed, that phospholipid is making bilayer. So this is phospholipid is making bilayer. Right now I'm making one layer, and then this is a bilayer. And after this, you're finding proteins are present. Depending upon the ease of extraction, if they are present outside only, we say peripheral protein. So protein can be peripheral, also known as extrinsic protein, because they can be easily extracted out. Sometimes the protein can be embedded inside. Okay, here right now I've done embeddation only half. They are embedded only half area. Sometimes what happens, they are embedded through and through. So making transmembrane protein. This is the way they are making transmembrane protein. Both are integral. So integral proteins can be half embedded or they can be fully embedded. When they are fully embedded, that time we call it as transmembrane protein. So these two both are the integral protein. And this one, only this one especially, this is known as transmembrane protein. So I hope this much is clear to you. The cell membrane is made up of phospholipid bilayer with proteins which can be extrinsic or which can be transmembrane also. Now attached to this protein, oligosaccharide is there, carbohydrate. If it is attached, we say it is known as glycoprotein. Attached to this, it is known as glycoprotein. Or sometimes oligosaccharide is attached to the lipid. We call such condition as glycolipid. Up till here concept is clear. Now, apart from the phospholipid, protein and oligosaccharide, now the fourth component which is present in the cell membrane is cholesterol. So cholesterol is present here in this way. Before this, let me explain you the structure of cholesterol so that you can understand. Now see beta, what is cholesterol? Try to focus. This is the diagram of cholesterol. Okay. And see here in this cholesterol, if you find this OH group, Please focus, this OH part is only hydrophilic and remaining part is hydrophobic. Remaining part is hydrophobic. Please focus carefully. Only this OH part is hydrophilic, remaining part is hydrophobic. Now see cholesterol. Cholesterol is a type of a lipid, but it is not fat or oil. It is actually belonging to another category which is known as steroids. And most of the cholesterol molecule is hydrophobic. Because of this, it is attracted to the hydrophobic hydrocarbon tails. Okay, it will be attracted towards the tail. But one end of the cholesterol is having OH group, which is hydrophilic, and it is attracted towards the phosphate head. Cholesterol molecules are therefore positioned between the phospholipid in the membrane. So they are placed in between the phospholipid. Is it clear, beta? Now, see one thing, our model is fluid mosaic model. Why we say fluid? Fluid means it should be neither too solid and neither too liquid. Fluidity is determined by the phospholipid. If the fatty acids are rich in saturated hydrocarbon, the membrane will be less fluid. So to make the membrane fluid, we have to make it rich in the unsaturated fatty acid. And we should use cis type of fatty acid. Is it clear? Instead of using the trans fat. We should not use trans fat, we should use cis fatty acid. If suppose the temperature is increasing, the fluidity can increase. And if temperature decreases, the fluidity will decrease. And we want our membrane should be fluid, whatever temperature range will be there. Okay, reason is that the cell membrane should do endocytosis and exocytosis very easily. So to maintain the fluidity in the extreme conditions of temperature, cholesterol plays very important role. Cholesterol will enter inside between the tails and will make them fluid in the low temperature. Try to understand, when the temperature is low, I think so membrane fluidity will decrease. Okay, when the temperature is low, membrane fluidity will decrease. So to increase the fluidity, I think so I should increase the concentration of phospholipid so that the membrane phospholipid tails can be separated apart, so that they should not go for freezing very early. Is it clear? Let me write for you once again, try to understand. Now see beta, try to understand. If suppose your unsaturated fatty acids are there, check in this first diagram. This is with respect to unsaturated fatty acid. Okay, beta? So when more unsaturated fatty acids are there, then you can see the membrane is fluid. Why fluid? Because the unsaturated fatty acids in the tail, they are having the double bond. So Nick are coming here. So they are preventing packing, and as a result, they are increasing membrane fluidity. But if suppose the fatty acids are rich in saturated hydrocarbons, then they are tightly packed. In the tightly packed conditions, the chances are fluidity will decrease. Understood up to here? Now, what is the role of the cholesterol? See. Now, see what is the role of cholesterol? Cholesterol will reduce membrane fluidity at moderate temperature by reducing phospholipid movement. Okay, so when the temperature is high, it will not allow the phospholipid to move easily. But at low temperature, it will not allow to go for solidification also. So it is working as a buffer. It does not allow the to fluidity, neither to viscosity. And check in the diagram, if you see carefully, this is the cholesterol hydrophilic part. Clear, beta? This is cholesterol hydrophilic part, which is attached to the head, and remaining part is embedded inside in the tail part. So cholesterol is working as a buffer, preventing the movement of phospholipid at high temperature, and at low temperature, it prevents its solidification. Now they have given us the phospholipid movement to try to understand what type of movement phospholipid is showing. Now see carefully. Focus the diagram carefully. If you see, this is the rotational movement shown by the phospholipid polar head. Clear? This is the lateral movement, side by side movement. So they can show rotational movement, they can show lateral movement also. Clear, beta? And if you see carefully, you are finding that they are showing the flip-flop movement also. It can show the transverse flip-flop movement also. Now, this is very, very important PMT question. Okay. Which movement is not shown by the protein? Protein being big in size. Protein are big in size, and that is why they cannot show flip-flop movement. They cannot show flip-flop movement. Please but understand it carefully. Phospholipid can show the rotational movement, it can show lateral movement, it can show flip-flop movement also. But flip-flop movement is also very rare in case of the phospholipid. Very rare they will show. Okay, but protein can never show flip-flop movement. It will be showing only lateral or rotational movement. And the movement of the protein over the lipid bilayer. How much protein are able to move over the lipid bilayer? Let me draw one diagram for your understanding. Try to understand, this is one protein I've drawn it. Okay, blue color, this is a protein. If this protein can move here and there very easily, I can immediately get an idea my membrane is very fluid. The fluidity is actually a measure. You can measure the fluidity of the lipid by the lateral movement of the protein. I hope up till here you are understanding the concept. Till now, let me revise what I explained you. I explained you that cell membrane is made up of phospholipid. It is made up of proteins. It is having cholesterol, and it is also having the carbohydrate. Now let us do the question practice. Try to identify what is X. X will be your polar or hydrophilic head, isn't it? And what are these by? What is your answer? You will give it is non-polar and hydrophobic tail. Clear, beta? And what answer you will give for Z? What is this? You will say this is phospholipid. So I hope this much labeling you have learned. Now let me give you another question also. Now see this question. Try to let us solve this question also. See, this question is given, we have to identify what is this U. So U is what? It is your phospholipid bilayer. Okay, so these are the options given, we have to match it. So I think so phospholipid bilayer will be U. Clear, beta? Then what is V? If you check, V is nothing, it's a protein. It is half embedded or fully embedded, this way it is there. So which type of protein it is? It is integral protein. So V is your integral protein. Clear, beta? And then what is W? It is peripheral protein, which is present only outside. So you will say this is peripheral protein. So this is W. Clear, beta? Then what is X? Try to identify, this is cholesterol. So X is cholesterol. So X is going cholesterol. Clear, beta? Then you do one thing, try to identify this also. See one thing, identify R. What is R? This is, I think so, sugar attached to the lipid. So R is your glycolipid. Then try to identify S. Here you are finding sugar is attached to the protein. So it is glycoprotein. Clear? And especially if you see carefully, this is, we can say it is basically they are emphasizing with the sugar. So it means, let us take this as a carbohydrate. We will write S for the carbohydrate. And this part, T, we can say it is a glycoprotein. Clear, beta? This is your glycoprotein. Now this one, Z. This is a protein which is fully embedded. So we will say transmembrane protein. So Z is transmembrane protein. Now sometimes the protein will work as a channel. We'll be studying in the next video. Channel means they are helping in the facilitated diffusion. Check in the diagram carefully, you're finding they need a channel, so that easily the diffusion can occur. For which type of substance? Which are hydrophilic, because they cannot pass the cell membrane easily, so they want the help And these type of protein which are giving the help so that the easily facilitated diffusion can occur. So they are known as channel protein. So Y will be your channel protein. So I hope today in this video, structure of the phospholipid bilayer, what is the role of all the molecules, and labeling, all the topic might be clear to you. Thanks for watching.